For a really long time, scientists knew that something inside ourselves was passing traits down from one generation to the next, but they had absolutely no idea what it was. This is the story of that hunt. It's basically a scientific detective story to find the chemical blueprint of life itself. And let me tell you, this investigation was full of some incredible twists and turns. For a long, long time, the scientific community had its eye on completely the wrong molecule. So today, we're going to follow the clues, look at the brilliant experiments, and see how they finally cracked one of biology's greatest cases. Our story kicks off way back in 1928 with a British bacteriologist named Frederick Griffith. Now, he wasn't even trying to find the genetic material, not at all. He was actually studying pneumonia. But what he stumbled upon was this totally bizarre phenomenon that would change the course of biology forever. So Griffith's experiment had two main players. Over on the left, you've got the R strain. It looks kind of rough and it's non-virulent, meaning it can't cause pneumonia. Then on the right, you have the deadly S strain, which has this smooth, protective coat that lets it hide from the immune system. So two strains of the exact same bacteria, one's harmless and the other is a killer. Okay, so Griffith starts with a simple control test. He injects a mouse with the live, harmless R strain, and, just like you'd expect, the mouse is perfectly fine. Its immune system takes care of the bacteria, no problem. Next up, the deadly S strain. He injects this into another mouse, and the result is also unfortunately predictable. The mouse gets pneumonia and dies. So yep, the S strain is definitely the bad guy here. Now, this is where it starts to get really interesting. Griffith takes that deadly S strain bacteria and kills it with heat. When he injects these now dead bacteria into a mouse, the mouse lives. So the key takeaway here is the bacteria have to be alive to actually cause the disease. And this is the part that baffled everyone. Griffith then mixed the harmless live R strain with the harmless heat-killed S strain, and he injected that combination into a mouse. Now, logic would say the mouse should be totally fine, right? Two harmless things can't make a deadly thing? Wrong. The mouse died. And when Griffith looked at its blood, he found living, deadly S strain bacteria. So how in the world was this possible? Griffith figured that some kind of chemical substance, something he called a transforming principle, must have passed from the dead S strain cells over to the live R strain cells, and it permanently changed them into the deadly S type. He had seen this incredible transformation, but he had no clue what this mystery agent actually was. The first major clue had just been uncovered. So, the race was on to figure out what this transforming principle was. At the time, scientists had two main suspects for the molecule of heredity. You had the really complex and versatile protein, and then you had this seemingly simple nucleic acid, DNA. The central question was crystal clear. Which one of these molecules was carrying the genetic instructions to turn harmless bacteria into killers? The entire scientific community was basically asking, who done it? You know, at the time, protein was the odds-on favorite. I mean, it made sense. Proteins are incredibly complex, they're built from 20 different amino acids, and they do a million different jobs in the cell. DNA, on the other hand, it's just built from 4 basic units, these nucleotides. It just seemed way too simple to hold the massive amount of information needed to build an entire organism. It was kind of like comparing a whole encyclopedia to an alphabet with only 4 letters. Then, in 1944, a team led by a guy named Oswald Avery decided to solve this mystery with a really brilliant process of elimination. Their plan was simple. Just systematically destroy each suspect, one by one, and see if the transformation still happened. Okay, so here's how their amazing method worked. They took the juice from the heat-killed S strain bacteria, and they treated it with different enzymes. First, they used an enzyme that just destroys RNA. When they mixed this with the R strain, yep, transformation still happened. Okay, so RNA was cleared. Next, they destroy the protein. And again, the transformation still occurred, so protein was also off the hook. But then they used an enzyme that specifically destroys DNA. And bam, the transformation stopped. Completely. The mystery was solved. The transforming principle had to be DNA. Now, you'd think that would be the end of it, right? Case closed. But a lot of scientists were still really skeptical. The idea that protein was the genetic material was just so ingrained in their thinking that they demanded more proof. The case needed one final, definitive, smoking gun experiment to convince everybody. The perfect tool for this final test came in the form of a bacteriophage. which is a virus that infects bacteria. And why was it so perfect for the job? Because it's incredibly simple. It's basically just a protein coat on the outside with its genetic material, DNA, tucked inside. When it infects a cell, it just injects its genetic material to take over. So in 1952, Alfred Hershey and Martha Chase designed this just beautiful experiment using radioactive isotopes as tags. It's so clever. See, protein contains sulfur, but not phosphorus. So they tagged the protein coat with radioactive sulfur-35. And DNA contains phosphorus, but not sulfur. So they tagged the DNA core with radioactive phosphorus-32. This gave them 2 batches of viruses, one with a hot protein coat and one with hot DNA. All right, next, they let each batch of these phages infect some bacteria. After a few minutes, they put the whole mixture in a kitchen blender, and yes, I mean an actual blender, to literally knock the empty virus coats off the outside of the bacterial cells. The whole point was to separate what went in the cell from what stayed out. After the blender, they spun the samples in a centrifuge. The heavier bacteria all collected in a little pellet at the bottom, while the lighter virus coats stayed floating in the liquid up top. So the whole experiment came down to one critical question. Where is the radioactivity? Is it in the pellet with the bacteria, or is it in the liquid with the empty virus coats? The answer to that single question would finally settle the debate, and it would close the case on the identity of the genetic material once and for all. And the results were just unmistakable. In the batch where they had radio-labeled the protein, all the radioactivity was found in the liquid on top, meaning the protein coat stayed outside the bacteria. But in the batch with the radio-labeled DNA, the radioactivity was found down in the pellet. The DNA had been injected inside the bacteria. The conclusion was just undeniable. Since the DNA went into the cell to start the infection, while the protein just stayed outside, DNA had to be the genetic material. This was the smoking gun. Unequivocal proof, dated 1952. The case was finally closed. So let's just quickly recap this incredible scientific journey. It all started back in 1928 with Griffith discovering some mysterious transformation. Then, 16 years later, in 1944, Avery's team pretty much proved the agent was DNA. And finally, in 1952, Hershey and Chase delivered the final knockout punch. Over 24 years, science had followed the clues to unlock one of the biggest secrets of life. And that discovery, it really paved the way for everything we know about genetics today. But it does leave us with one last little thought. In biology, there's very often an exception to every rule. So is DNA always the carrier of heredity? Well, it turns out no. Some viruses, like the one that infects tobacco plants, actually use RNA instead. But for the vast, vast majority of life on Earth, from tiny bacteria all the way up to us, DNA is the chosen and now proven hard drive for our genetic code.